Cofactors. Understand the importance of cofactors in enzyme action. We will also clear our concept on the types of cofactors. A human body consists of numerous cells. These cells are considered as functional units of life. Each cell encloses a centrally located nucleus and cytoplasm. The cytoplasm has various organic molecules like enzymes suspended in it. Some of these enzymes are inactive, that is, they cannot start a reaction process themselves, while some enzymes are active, which do not require any additional aid and bind to the specific substrate molecule and break it into the product molecules. But in the case of certain inactive enzymes, also termed as apoenzymes, an additional component generally known as the cofactor is required to activate them. The cofactors can be of two types: inorganic metal ions or organic compounds. Generally, an organic compound which acts as a cofactor is termed as a coenzyme. Let us study the role of heme group as a coenzyme. When the heme group binds loosely to the peroxidase enzyme, it forms a part of the active site. This complex formed results in a catalytically active peroxidase enzyme. So, when the substrate molecule of hydrogen peroxide attaches to the active site of this enzyme, it breaks up into water and oxygen. Let us draw our attention towards the role of inorganic metal ions, which also act as cofactors. Taking zinc as a cofactor for carboxypeptidase enzyme molecule, let us study its role in enzyme activation. Zinc ion is attached tightly to the enzyme, forming a part of the active site. Such metal ions which are bound tightly or covalently to the enzyme are known as the prosthetic group. This activated enzyme acts on a substrate molecule and breaks it into a product molecule. If the zinc molecule, that is, the cofactor, is removed, the enzyme loses its catalytic activity and cannot act on the substrate molecule. Thus, we conclude that the cofactors play a vital role in activating the enzyme. To summarize, the enzymes are of two types: active and inactive. The active enzyme directly acts on the substrate and converts it into the product molecules. The second type of enzyme is the inactive enzyme, which requires a cofactor for activation. These cofactors can be organic compounds, generally termed coenzymes, or can be metallic ions.